Good morning and welcome back to the Extraordinary Technology Conference 2017. This morning it is my pleasure to introduce another second and third year speaker, and Dr. Glen Rein is going to be speaking on the memory of water. He holds a PhD in neurochem from the University of London, and he's been a researcher for 30 years in a vast array of different fields with a vast array of experience and knowledge. He's also the founder of the Quantum Biology Research Lab. Please welcome Glen Rein.
All right, thank you. Thank you, everybody. And I'm going to talk today about water, although I think I'm in the biology section, so technically I'm talking about the biology work that I've done in the past tomorrow at Tom Vallone's conference. But today I'm going to focus on water. After all, the body is like 80% water, right? So we can make believe it's a biology talk. but water is so fundamental and has this amazing anomalous properties about storage of information that I'm going to focus on today. And I want to start with the parallels between these two molecules. As you can see on the bottom, their molecular structures are quite similar. And in fact, the silicon dioxide molecule is obviously been utilized and discovered in the electronics industry, that we know so much about SiO2, and in contrast we knew so little about H2O, despite the fact that the body is 80% water, etc. And what's particularly interesting in both of these molecules is their quantum properties, and particularly coherence, quantum tunneling, and quantum resonance. But the fact that the water molecule itself and its quantum properties and its anomalous properties are not really studied by the medical profession. But it's certainly studied in nature. I mean, nature, obviously there's a lot of water on the planet as well, and water on the planet is structured. And it's structured just naturally by the movement of the water over the rocks and et cetera. And of course, everyone here knows about Schauberger and his pioneering research in this field. But the main point here is that the living water that's found in nature has amazing properties. And particularly, I'm going to talk a little bit about LOURDES water, which is the natural springs water in France, where people have been going for literally thousands of years to get healed. So, I mean, there's psychosomatic effects, and then there's real effects. And people obviously have been getting better, or they wouldn't keep going back for thousands of years. So LOURDES water is just one of many natural springs throughout the whole planet. And a colleague of mine, Lee Lorenzen, has actually studied this water extensively and discovered that it has some very unique properties. In particular, he measured the O17 NMR spectroscopy of LOURDES water and other natural healing spring waters, and discovered that there's a resonance frequency at 65 hertz. And this is apparently the signature that is unique to these healing waters, and it's the signature that apparently gives the water its healing properties. So Lee decided that he was on a mission to figure out how to generate structured water through a machine that would generate the 65 hertz NMR peak. And then lo and behold, his water had a lot of healing properties. That same water also has a hexagonal structure, and in contrast to the water on the left, which is more typical of the water that surrounds molecules and ions and electrolytes in the body. The problem is that these particularly large structures are so large, they don't get into the cell, they don't cross the cell membrane, so that the efficiency of absorption of minerals and ions into the cells in our body is remarkably inefficient because of these huge structures and clusters around each molecule. However, in the case of hexagonal water, this very small structure can readily pass through the cell membrane. Here's a comparison of the NMR signature I was talking about in different kinds of waters. And you can see that highly contaminated water, including the tap water that we drink, have on the y-axis here we have actual hertz value of the NMR peak. So as I said, well, we could start backwards. As I said, that Vivo water, Lee's water, which is, by the way, commercially available, has a value of about 65. Saline and glacier water are similar. Plasma is a little bit higher. And then when you get to even distilled water, contaminated water, and tap water, all have values way over 140 hertz. So this is a particularly beneficial frequency, as it turns out. And after structuring the water in a rather unique way, Vivo water has this characteristic peak, and it's stable. It lasts for about 2 years. So in contrast to LOURDES water, which is worth mentioning, that water has to be drunk right on the spot. If you take the water back to your hotel, that NMR signature fades in about an hour. But that's why people drink it right on the spot. Okay, so now let's focus now on what kinds of electromagnetic field treatments we can use to structure water and to treat water and to inform water with specific frequency information. This all started back in the 80s, I believe it was, when bioelectromagnetics researchers who've been studying the biological effects of electromagnetic fields for hundreds of years, a Russian scientist actually said, well, maybe, all cells are surrounded by water, so maybe the electromagnetic fields are interacting with the water and primarily, and then the water transfers the information to the cells. So to test that hypothesis, they treated water with an electromagnetic field and then added that water to the cells in culture, and they got basically the same effects as directly treating a cell, like in tissue culture, with an electromagnetic field. So that kind of opened up the whole Pandora's box about water memory, because when they treated water with an electromagnetic field, they discovered that the effect lasts for weeks. And the dogma and the theory says, well, if you turn on the signal and you change the properties of water, as soon as you turn off the electromagnetic signal, the properties should dissolve and go back to normal. But that's not the case. And apparently this phenomena of water memory is an anomaly, shall we say, and it doesn't really obey the standard laws of physics. Hence, it has not really been accepted in mainstream biology. Now, alternative scientists have been studying this phenomena for a long time, and there are now scientific publications, but the mechanism is really unknown. It's still unknown. We don't know exactly how and where the information is stored, and we don't know how biological systems can read the information, because it's not a molecule. Remember, you can actually do this with pure distilled water, and there are no molecules present. So we're not talking about a molecule binding to a receptor in a cell. We're talking about water having some kind of an effect on the cell, assumedly not through the ordinary molecular receptors. And furthermore, this phenomena of reading information from water applies to mammalian systems, bacterial systems, even yeast and microorganisms and plants. So it's kind of a universal phenomena, and we really have no clue about how it works. That should qualify for the anomalous behavior and esoteric properties of water, which fits right in with this conference, right? So there are many different kinds of energy fields which have been used to treat water. Obviously, the original experiments were done with magnetic, and even with a DC magnet, you can do similar effects. And in addition to electromagnetic fields, you can also treat water with scalar, or what I call non-classical electromagnetic fields, and I will show some data on that, in addition to light and sound energies. So water seems to have this universal property. It doesn't really matter what kind of energy it is. It'll just soak it right up and store the information. What happens when you treat water with these kind of energies? Well, these are a list of some of the physical properties, physical chemical properties that have been reported in the scientific literature. And I want to distinguish here between these kind of chemical effects and physical effects as distinct from structuring of the water. And people get confused on that, but apparently electromagnetic fields can also cause the water to structure, as I showed you in the earlier slides, in hexagonal or pentagonal, actually in many different kinds of structures depending on the nature of the energy. And in this particular lecture, I'm going to focus on the electrical properties and the optical properties of water and how it changes. And the molecular information. Now, this is actually technically the title of the talk that you have in your program, and it has to do with the fact that molecular information. Now, this is energy associated with a molecule. I'm not talking about an electromagnetic field generated from a machine, but an electromagnetic field generated from a molecule. You know, every molecule has an electromagnetic field around it. The dogma is that it's so weak that it doesn't really do anything. When a molecule binds to a receptor, the dogma is that it's the molecule itself which triggers the biological response. But there's now information that that electromagnetic field around the molecule can also have an effect on biological systems. So what we're talking about here is literally transferring a chemical molecule from a solution into pure water. And this water then acquires the same biological properties as this water. This was originally done by Jacques Benveniste in France, and he actually got this published in the prestigious journal Nature, but in fact, it was retracted shortly after because the scientific community freaked out and said, "What do you? This is magic. This is voodoo. You know, there's no science behind it. We don't understand how it works, and you can't publish an article like that." So they retracted it. But Benveniste coined the term digital biology, referring to the fact that digital information from the electromagnetic fields of the molecules can have all kinds of biological effects. More recently, this kind of research has been taken up by another French scientist by the name of Luc Montagnier, who happens to be a Nobel Prize winner. He discovered the HIV virus and got awarded the Nobel Prize, and now he is obsessed is probably a good word, but he's really shifted all of his research focuses on this phenomena, and he's working with a particularly interesting molecule called DNA, and which is a rather complex molecule, but nonetheless he can transfer that molecule into water. This water, which has been measured, no DNA in the molecule. You give this water to PCR machine, and the machine which duplicates DNA, and the machine starts duplicating the DNA as if there were real molecular DNA in the water. The machine thinks that there's DNA in the water and starts to replicate it. So it's a real phenomena. He's published it as well, and with his name, he's kind of advancing the whole field of molecular transfer. Now, I was going to talk about some of my own research in this area, and I'll just briefly summarize that, so it's technically the title of the talk. I did a similar experiment, but I used a drug in this case for the chemical. The drug was a MAO inhibitor. MAO is an enzyme, monoamine oxidase, and the drug MAO inhibitors are used as antidepressants. And in my experiment, this little arrow was a laser beam, in contrast to the two previous scientists I was just telling you about, who used basically an electromagnetic field with the Schumann resonance to transfer or carry the information from here to here. In my case, I used a laser. And I didn't treat water in these experiments. I treated cells directly. Of course, they're surrounded by water. So in essence, the water was receiving the information. But the cells, nerve cells grown in tissue culture, were exposed to the laser by itself, and the laser modulated with molecular information of the MAO inhibitor. The laser had no effect on the cells, but the laser modulated with the information did affect the DNA, the enzyme MAO. However, there was not an inhibition, but there was the stimulation. So this drug was supposed to inhibit the enzyme, but the information from the drug actually stimulated the enzyme. So what can I say? It was certainly not a psychosomatic experimenter effect, because I was thinking the results would be the opposite. And it also tells us that the way that that information is coupled to the laser is critical. I didn't actually shine the laser beam through the drug. I shined the laser beam just over the drug was like a powder sitting on the surface, and I shined the laser beam. Gee, I happen to have a laser beam right here. Cool. I shined the laser beam right over the top of the drug. So the laser picked up the information from the drug and carried it into the cells. Okay, so that's the title of the talk that's in the flyer that you've been handed out. So now I'll continue with the other talk, which is mostly about information storage. How can water do this miraculous phenomena? This science is, as I said, it's not really published, but it's just starting to be published. Here's an article by Cyril Smith that was published and addresses not only the fact that electromagnetic fields can store information, but the A field, or the magnetic vector potential, can also do that. And you guys all know about A fields and scalar energy, which is another term used to describe this kind of subtle energy which is present when you cancel the electromagnetic field, simply put, by opposing the two electromagnetic fields 180 degrees. They cancel, and the energy that's underneath is not a force field anymore, it's a potential field. And it's called the magnetic vector potential, as I said, and it has lots of different names. And you guys are all experts on that, so I don't have to address that any further. I want to talk about the mechanism, exactly how this occurs at the molecular level. And the standard dogma for the last 20 years was that this hydrogen bond structure cluster that I showed you earlier around an ion somehow or other traps the information and allows the information to be stable. The problem with this model is that these structures don't last very long. They last for picoseconds, and they constantly form and break apart and then reform and break apart. So any information that would be stored in this kind of a structure would be dissipated and lost into the bulk water. So although these kind of structures have very interesting quantum properties, electron tunneling, delocalized pi electrons, and all that really cool stuff, which makes it very enticing to think of a theory based on this molecular structure held together by the hydrogen bond, it doesn't really make sense to me. And then of course there's the complicated fact that a lot of the information that we're talking about is quantum information. So we have the biologists who have no idea what's going on. They see this phenomena and they go, well, okay, that's interesting, and we have no idea. Then we have the physicists talking about information storage in semiconductor, in liquid crystals, in the electronic circuitry. So that's okay. And then now they take that basic observation and extend it into the quantum domain and talk about qubits of information and quantum information as a new kind of information which can also be stored in water, because water itself is a quantum system. So now the situation is getting even more complicated, and now most people throwing up their hands and say, well, we have no idea how information is stored in water. Until Emilio del Giudice comes along, an Italian theoretical physicist who comes along and says, well, okay, let's talk about not clusters like I've been talking about so far in this lecture, which all are mediated and held together by short-term electrostatic hydrogen bonds. The hydrogen bond is an electrostatic bond between a positive and negative charge. So Del Giudice says, okay, I want to talk about what he calls quantum domains, or coherent domains, CD, coherent domains, which are connected by long-range electromagnetic fields. And these structures form new configurations with a minimal energy which makes them stable. So we are talking about a structure which is about the size, 75 nanometers is the estimated size of these molecules. So we're not talking of these structures. They're not molecules. It's a quantum structure, if you will. And they're held together by the molecular oscillations which allow a coordinated, coherent kind of communication between the individual molecules. And furthermore, that it's the resonances between the ground states of these molecules that allow this structure to be held together. And of course, the ground state, as this group knows, is really the link to the vacuum energy domain. And in contrast to the electromagnetic world, where the excited state of the electron is what's considered and most relevant, here we're talking about ground state effects and resonances between ground states. So anyway, so we have this kind of configuration of molecules which traps the electromagnetic field inside. Now, the ordinary electromagnetic fields, the classical electromagnetic fields, are excluded, and the kinds of electromagnetic field that gets trapped inside of this configuration of molecules is referred to by Del Giudice as non-Maxwellian. Well, that's another word for non-Hertzian, which is a familiar term in this audience. So in a way, we're talking about a type of scalar energy which is trapped inside these domains, acquires quantum properties, and stores information. And that is now the best theory about how water is stored, information is stored in water. I mean, there are other theories, like bubbles, microbubbles is another theory that's out there, but I particularly like this theory because it accounts for the quantum properties of the information transfer. Okay, so now I want to talk about some original experiments. How much more time do I have? Do I have any kind of a guide here about time? No, I'm just going to keep talking, huh? All right, at some point the hook will come along and I'll cut it off. So I want to talk about some original experiments that I've done. And here's a picture of me in my lab from some previous incarnation. And to be quite honest, my lab looks pretty much like that these days too, except my hair is a little longer. Okay, so I'm going to start with a method that I've used called ultraviolet spectroscopy to measure the information in water. And this is a method actually that Marcel Vogel discovered pretty much the same time that I did. We were working in parallel. We were both in the San Francisco area, and we both discovered this phenomena that ultraviolet is a kind of light optical spectroscopy that is used a lot, but not for this particular application. But the catch-22 is you have to work at around 200 nanometers, which is really the far end of the ultraviolet. And most UV experiments are done like 300 and 400 nanometers. And of course, if you keep going higher, you eventually get into visible light. So this is a kind of novel region to be doing these experiments, but that's where the effects are seen. And in order to do these experiments, you need a research-grade spectrophotometer because the cheap ones on the bench top don't go down that low. And what you're basically measuring here is the excited states of the electrons in the dissolved oxygen in the water. Now, oxygen itself is very interesting because it also carries information, and that's another whole lecture, but it was done by a nurse who did this research many years ago and discovered that healers could actually affect hemoglobin in the blood, and hemoglobin carries oxygen, so she postulated that oxygen carries healing information. Anyway, so that's a little background on UV spectroscopy. So here's a typical UV spectra. And the x-axis, you've got wavelengths, and these are around 200 to 230 nanometers between here and here. And this is just the absorption of light at that particular frequency. So the first thing you got to do to be scientific about it is to measure 3 controls in a row, and you could see they're literally superimposed. So it's a very robust phenomena, and you really basically get this same thing no matter what and where and when you do the measurements. So the first thing I was curious about is, okay, thank you. I have a 30/20 minute, one of the two, time frame, so we're good. What happens to water when you vortex it? And there's a lot of esoteric teachings that say, you know, you structure the water when you vortex it and it acquires unusual properties. So here's an example of what happens. And you can see that at the peak, there's not a lot happening, but it's more of a shoulder effect, and the effect is seen at around 230 nanometers, but there's clearly a difference. So now I'm going to show you a whole bunch of different slides of pictures of different kinds of patterns when water is exposed to different kinds of energies. Okay, so here's the first experiment that I want to talk about. I use this from a talk I gave in Germany, and that's how they spell SKALAR. Sorry. Whoops. Jumping ahead of myself. So in this particular experiment, I wanted to see, well, first of all, if there was an effect. And there's a huge effect around this between the control, which varies, but when you treat it with the treatment was a column that had magnets on either side, north against north, and then you just pour the water down the center. It goes by very quickly by the field that's created between north poles opposing each other, and that's a kind of DC scalar electromagnetic field. And the particular effect, it was to decrease the ultraviolet spectroscopy. As you'll see, sometimes you get an increase and sometimes you get a decrease. And then I was asking the question, well, if you just keep it on the bench, how long does it last? And I did it in a shielded and a non-shielded environment. So this is basically in the ambient air. And the sample of water was put in a mu metal cage to protect it from external electromagnetic fields. And you could see that, the non-shielded effect goes away in just a couple of days, and already here, this is already, sorry, no scales, about a week. It lasts for about a week in this particular experiment, demonstrating the difference between shielding and non-shielding. Okay, so here's another experiment that I did with a classical electromagnetic field done at 42.7 kilohertz, and I'm sure that a lot of the people in this audience know the Keeley frequency, which is apparently a frequency which disrupts water and causes all kinds of bizarre anomalous behavior. So according to Keeley, so I thought this would be a good frequency to stimulate the water, get it excited, and then see what kind of effects I can measure. So you can see here that the effect is quite, well, it's a measurable effect. It's small, but it only occurs at the peak, and there's no shoulder effect. In contrast, you'll see, when I do the same kind of experiment with scalar energy from a caduceus coil, which is, this was actually done at a different frequency, but you can see at this particular frequency, the non-classical electromagnetic fields also had the same kind of effect here, but in addition had a shoulder effect. And in this case, you can see it's a decrease relative to the control. Okay. Next was a similar experiment, but in this case, it wasn't a single frequency. I scanned the frequencies from hertz into the kilohertz region, and treated the water for 20 minutes, take these measurements, usually a short time afterwards, just because I don't know how long it's going to last. It's a different question, obviously. And you can see that, again, there was a much larger effect at the peak, and in this case, the shoulder effect was an increase, which had a particular peak right around here, about 220 nanometers. So it's the morphology of this peak that we're interested, which obviously changes depending on the kind of energy that we're using to treat the water. And here's an example of a laser, a very weak 5 milliwatt laser, 30-minute treatment, and in this case there was no effect at the peak, a shoulder effect in a positive direction, in contrast to the previous slides. In summary, the kind of effects that I see are threefold. One, where you affect the peak and not the shoulder and not the peak, and the shoulder, and you can have the peak and not the shoulder. You can have all kinds of variations on the theme, and the place where the maximum effect is observed, here would be about 220 nanometers, and here we're talking about 240 nanometers. So that's a summary of all of the different kinds of energies, and I've done this with non-classical electromagnetic fields, classical electromagnetic fields, and bioenergy from a healer. And that's like another whole lecture, maybe next year. Okay. Oh, here's another kind of energy. The energy emitted from a geometric pattern. Believe it or not, these three different geometric patterns generate different electromagnetic fields which can actually be measured by placing a sample of water on top of the pattern. Of course, in this case, you got to keep it there for 24 hours because the field is very weak, obviously, but it does have an effect on water. And here's the UV spectroscopy data. In this case, I'm actually showing you numbers. Let's be scientific about this, right? And here, this is on the y-axis, we have the difference between the control and the treated. So in the absence of an effect, you'd probably see, like in the control, it would fluctuate around plus or minus 1. The snowflake pattern caused a decrease. This was yet another treatment. The triangles and the golden mean spiral caused an increase. And again, these are the patterns. That's the triangles, and that's the golden mean spiral. And indicating that what visually looks like, is more or less similar, these two things have a more or less similar pattern, are causing, that's the snowflake, and that's the triangles. The snowflake and the triangles have opposite effect on the water. So there's interesting phenomena here, and obviously it needs a lot of further research. I have done some research with three-dimensional geometric structures made by Greg Hogue, and I've also seen similar effects. Okay, so now we're going to focus on the scalar component, which I've talked about a little bit, and I want to describe a particular experiment where I compared the classical and non-classical electromagnetic fields using a solenoid and a bifilar coil. Okay, so all the engineers in the audience can relate to this, I'm sure. In the bifilar mode, the current flows in opposite directions. In the ordinary solenoid coil, the current flows in parallel directions. So we have two different coils, and they have, obviously different properties. And what I did was to, well, actually, I didn't do this. It was Bill Tiller who collaborated with me on this particular project, who calculated the strength of the B field in these two different coils. And so what we did was we adjusted the intensity of the, this is incorrect, sorry, the amount of current flowing through the coils was adjusted so the total output of the two coils was the same. Okay, after doing that, Bill calculated the intensity of the magnetic field. So this is -3 Tesla, which you would expect is a reasonable field present from a standard solenoid coil. But in the bifilar situation, the strength of the magnetic field was negligible, 10 to the -12 Tesla. So in essence, there's no magnetic field present, but there is a very weak A field present, which is the magnetic vector potential, which he calculated as well. So here's the experimental setup, and the actual results are quite interesting. Okay, now again, let's see, in this particular graph, we're measuring the absorption at one particular wavelength, and I'm calculating the percent change relative to the control. Not the delta, but the percent change. And again, you could see with a classical electromagnetic field, these 6 different experiments, sometimes it goes up and sometimes it goes down. And if you remember the standard control, the control would be 0 basically, or pretty close to 0 in terms of the percent change. In contrast, when you do the same experiment with a scalar field, from the same frequency and the same strength, you get a completely opposite effect. And in these 6 experiments, it was all negative compared to the control. It went down relative to the control. As I showed you earlier, happens a lot in both directions. So here's a pretty definitive conclusion about the difference between classical and non-classical fields, same frequency and same strength. Okay, so now I'm going to switch gears to a different method of measuring the properties of water. In this case, I want to talk about a technique called nonlinear dielectric spectroscopy. And since most people may not know what that is, I thought I should talk a little bit about the electrochemical properties of water, because that's what we're really measuring here, is the electrical properties of water. And it turns out that there's a lot of science about this. Water is clearly a nonlinear system, and the theoretical modeling of this phenomena has been based on systems theory, chaos theory, bifurcation theory, and et cetera. So there's a lot of mathematics behind this and modeling. But the experimental observations are that you can induce electrochemical oscillations in water by applying the appropriate stimulus, and that these oscillations are long-lived, and they vary according to the experimental parameters, and they describe this behavior as non-ohmic. In other words, non-electrical, as in ordinary electrical behavior was described by Ohm, and that's why we use the term non-Maxwellian and non-Hertzian to describe the nature of the fields that we're applying to the water. But here we're talking about the electrical properties of water as being non-ohmic. I thought that was kind of interesting. Okay, so what you're basically doing here is stimulating the system with applied voltage, and then you measure the induced current. Okay, and the system can go either in 2 directions. It can either go toward a stationary steady state equilibrium state, or it can go to an oscillatory state, which is in non-equilibrium and unstable, and can be modeled in terms of a bifurcation. And what I'm doing is trying to generate this state experimentally and take measurements of this unstable non-equilibrium state. So here's the standard electrochemical equations that they use. They're called current voltage curves. You apply the voltage, you measure the current, and it's nonlinear in the sense that it both negative and positive. And here's the equations to describe the resistance or impedance associated with the flow of current through the water. And here's the kind of results they get with water at different excitation intensities, the voltage, and you get different dispersion curves depending on the experimental parameters that you use to excite the water. But in my particular case, I'm not so interested in the actual dispersion. I'm interested in these oscillations. So here's an example that shows that under certain voltage excitations, you get these induced oscillations, but at lower voltages and higher voltages, you don't. So this is the phenomenon that I'm interested in, because if you do a frequency analysis of these oscillations, you can get frequency information about the water. And here's an example of that. There's an actual spectra, which means frequency versus the amplitude of at a particular frequency. And you can see that this particular frequency would correspond to a resonance frequency of the water. So that's actually what we're measuring here with this particular device. And what's really cool, since it really measures resistance or impedance, you can actually get a real and imaginary components. So you get a spectra of imaginary impedance as a function of frequency, which you can see is very different from the ordinary impedance measurements that most scientists use. I got to say, as excited as I was when I saw that, I thought, okay, you know, all this scalar stuff, all this subtle energy that I'm putting in the water, this is good. I can use this new method of measuring the information in the water. Well, it turns out not really. The interesting effects were all seen here. Sorry. That's science for you, right? Okay, so I've taken the standard procedure and modified it, and I call it resonance frequency spectroscopy. I mean, you got to give it a name, right? Hey, I could have named it the Glen effect or something, right? But I thought I'd give it a nice scientific name. And it is, just like I said, I'm using frequencies in the range of 1 to 100 kilohertz because the machine that I have only works in these regions. And the frequency response is the induced current. And now the induced current response is complicated. It varies in shape, magnitude, polarity, and time, depending on the nature of the stimulus and the nature of the energy we're putting in the water. Now, when I say time, what I mean is this phenomena called frequency hopping. I mean, you get a response at a particular frequency, and you do the experiment again, and you get the response, but at a slightly different frequency, or higher or lower. And that phenomena is published in the literature, and it's called frequency hopping. The frequency doesn't stay the same. It hops around one particular value. So this makes it a little challenging to get quantitative data from this, but what you just have to do is, instead of measuring the intensity of the response, intensity of the induced current response, what I measure is how often it appears at a particular frequency. So that way, if it moves, that's not considered a hit. It's not considered a response at that frequency. And that's how I present all the subsequent data. So here's an example of a typical spectra obtained over time, exciting the water with a 10 millivolt electric field, basically, at 562 hertz, and you repeat the measurement every 10 seconds, and then you get this response. This is the response. Oh, I see. What I'm showing you here is the response at one frequency. This is the frequency hopping phenomena. So this is done at one frequency, and the response that you get varies enormously over time. So this is the frequency hopping phenomena. Hence, I have to do the averaging as I just described. Okay, so let's look at some spectra in response to 7 minutes, in response to different kind of stimuli. Okay, well, here's playing music. So in all cases, the before and after, or the untreated and the treated. And you can see that in most cases, the treated increases. These are specific frequencies. Well, not really, they're numbers. But they occur at specific frequencies between 1 and 100 kilohertz. But you can also notice that in some cases, there's a decrease. So at some frequencies, there's a really large stimulus. Some frequencies, there's no effect at all, and other frequencies, you get a decrease. It's just the nature of the beast. It's so complicated. It's a complex system, and it's a frequency-dependent effect. And here's the kind of, this was classical music. Here's the effects of a healer. And in this particular case, again, you see, depending on the frequency, this was the difference between the control and the treated, the delta between the control and the treated. And you can see in some cases it's a positive effect, in some cases it's a negative effect. Here's the actual frequencies where you can see, and in most frequencies there's no effect at all. Okay, and well, we don't have time for this. Oh, yes, I do. I have time for this. I have, well, so then what I did was I took this one particular frequency, 2.75 hertz, and the device that I have allows me to take repeated measurements at that frequency. So this is repeat measurements over time. And if you do the measurements at that exact frequency, you get this kind of curve, which is pretty stable considering when you do the same experiment at 3.0 hertz instead of 2.75, you get this wild fluctuation. So indicating that if the system is stable at that frequency, that's some kind of a resonance frequency of the system. And of course you can then, this is a time domain curve, you can then get frequencies from this, but then it gets a little complicated, a frequency from a frequency, which is kind of bizarre, so I never did that. But that's just an example of the kind of experiments that you can do with this new method that I developed in my lab, and I have also been able to show that scalar energy can be distinguished from non-scalar energy using this kind of method, and that the method is reproducible, and it's a commercially available device, and if anybody really wants to repeat these experiments, come see me afterwards. So I appreciate your attention. Thank you very much.
Thank you very much, Dr. Glen Rein. Ladies and gentlemen, we're going to be taking a very short break and resetting some, excuse me, resetting the audio-visual.